Asia and other continents. Uh, Good morning, good afternoon, good evening for all. Let me share my slide for the presentation. How about is it visible now,
Yes, it's
Dr.
great.
Reem?
Yes, it is good.
Okay, thank you. My talk is on wheat blast, a potentially catastrophic disease threatening global food security. And I am very much thankful to Wheat Initiative for offering this opportunity to share the progresses of our work against a tiny fungal disease called wheat blast. First, I would like to introduce the map of Bangladesh. Bangladesh was affected by wheat blast in 2016. Here you can see Bangladesh is a South American country surrounded by India by three parts, and also we have border with Myanmar, and south part of the country is Bay of Bengal. The country is the most densely populated country in the world. Population is approximately 160 million. After Singapore and Hong Kong, Bangladesh is number one in the population density. And we have fertile land. Uh, and although land area is incredibly small, 147,000 square kilometers only, but it is the largest delta and highly fertile. We produce 3 crops per year. According to the world, data, Bangladesh is considered one of the most climate vulnerable countries in the world. And I am from a public university, Bangabandhu Sheikh Mujibur Rahman Agriculture University, and it was established in 1983 as an institute funded by JICA and USAID as a center of excellence in MS and PhD studies. And this institute was upgraded to Bangabandhu Sheikh Mujibur Rahman Agriculture University under the name of our father of the nation in 1998. In 2019, Institute of Biotechnology and Genetic Engineering was established, and I delivered 12 years struggle to establish this interesting institute with state-of-the-art research facilities. As I am going to talk about wheat blast, before I start my talk, I must remember and recognize the great scientists who have been working with me and supporting me to fight against this fungus. And the first I should remember Sophien Kamoun, Sophien Kamoun and Nick Talbot. They are from the Sainsbury Laboratory in UK. Professor Guliang Wang, Ohio State University. Professor Andreas Pontiedemann, University of Göttingen in Germany. Dr. Pankaj Bhaumik from National Research Council of Canada. Wang Zhongkang from the Chinese Academy of Science. And our collaborators also include Professor Yusuke Yamauchi from University of Queensland, Dr. Magdi Mahfouz from King Abdullah University of Science and Technology, Professor Yukio Tosha from Kobe University, and Dr. Haijun Gu from Institute of Crop Science, Chinese Academy of Sciences, and Dr. Daniel Kroll, University of Notre Dame in Switzerland. They significantly contributed, and the result I am going to present is the outcome of our collaborative effort. What I would like to tell you today, the story of the bad story, and one of the saddest story of the epidemic outbreak of wheat blast in Bangladesh in 2016, and how it impacted in our food security and economy. And we used the smartest field pathogenomics, open data sharing, and open science approaches to rapidly determine the origin of wheat blast in Bangladesh. And later, how to tackle the disease, we used CRISPR technology in rapid detection of wheat blast. We also tried to utilize the CRISPR genome editing and other frontier sciences for the mitigation of wheat blast. Why we need open data sharing and open science, I would like to share with you because it was a fantastic, successful experience when that plant health emergency came in Bangladesh. We utilized open data sharing and open science without or forgetting publishing a paper in Nature or Science. And I should remember Professor Sophien Kamoun, who first, shared his thought to use the open data sharing and open science. And it was the first approach in case of a plant disease. But before that, Ebola, Zika virus, and ongoing COVID-19 also used open data sharing and open science. And in that way, we can engage global scientific community to rapidly tackle any emergency utilizing the scientific skills and resources all over the world. Emergence of fungal diseases is increasing significantly in last 4 decades. If you see, it is increased fourfold. Though we modern agricultural practices, but diseases of major food crops cause up to 15% of pre-harvest losses. And in the human history, there are Irish potato famine and many other cases that created severe threat to human food and nutritional security. So disease is really a big challenge, and due to climate change is increasing. Now I would like to tell you the story of wheat blast. This report published in Daily Star in Bangladesh on March 1st in 2016 was the starting point. Sufyan shared this one through the Twitter as well as the Facebook, and we started discuss on this topic. And when we discussed, Sufyan and I, then I made this small post in Twitter and Facebook. We decided to determine the genetic identity and origin of the wheat blast because it has never been in Asia. And here is a brief summary of the outbreak, first outbreak of wheat blast. Here you can see 8 districts, 15,000 hectares of wheat was just devastated. Yield loss was up to 100%, and total 16% of that total wheat production was destroyed at that year. And we all know that wheat is one of the most important staple, providing 20% of the calories and 25% of the protein consumed by the human all over the world. And in Bangladesh, wheat is the second most important cereal crop after rice. Present demand is around 7 million ton, but our production is before the wheat blast, it was around 1.5 million ton. So we are heavily dependent on the global import. And later I shall tell you how a global import of wheat also caused the wheat blast. And demand is increasing every year with change in dietary habit and population increase. Bangladesh is now fifth highest wheat importing country in the world. And if we look at this graph, among the top 10 wheat producing country, China, India, very close to Bangladesh, and Pakistan is one of the top 10 wheat producing country. If we consider in total in Asia, more than 40% of the wheat are produced. And China is number one, India is second, as Bangladesh is very close to these two countries. There is a high risk of movement of this disease to those countries. Wheat blast before 2016, I had no idea. But when it came to Bangladesh, we checked the literature and we found that it is a fungal disease spread by a seed and crop residue or air, and it is caused by the specific pathotype, Triticum pathotype of Magnaporthe oryzae, causing the blast disease of more than 50 species of grasses. In Bangladesh, we have Magnaporthe oryzae in rice and other grasses, but wheat blast was in since 2016. Wheat blast was first discovered in Paraná state of Brazil in 1985, and then it spread to the neighboring countries, Argentina, Bolivia, Paraguay, and it was in South America. But in 2016 in Bangladesh, 2018 it moved to Zambia. I shall tell you later on. So Magnaporthe oryzae has lots of pathotypes or lineages, and they have limited, the host specificity. That means Triticum pathotype does not infect the rice. Rice one is oryzae pathotype. There are chances of, the host germ, Lolium pathotype in America, USA, Kentucky farm, one plant was infected, and they identified that from Lolium to wheat, but the story was there. And in Brazil, it was moved from the evolutionary story, how host germ was happened. We started our mission with Sophien, Diana Saunders from John Innes Center, Daniel Kroll. They significantly contributed in our wheat blast mission, and we hypothesized that wheat blast in Bangladesh may be caused by a native blast fungus from rice or other grasses through host germ, through mutation. Wheat blast in Bangladesh may be imported from South America through seed. So we had two hypotheses, and our approach was to use field pathogenomics for determination of the genetic identity and origin of the blast in Bangladesh. And here is the procedure. We collected the samples and then extracted the RNA and did the RNA-seq analysis. And field pathogenomics is a new method to analyze fungal disease from field samples and pinpoint the exact genotype in a faster way rather than the classical culture and isolation method. Because culture and isolation method, wheat blast and rice blast morphology, it's same, difficult to distinguish. It was our protocol. We extracted the total RNA and then sequenced RNA-seq and assembly by Trinity software. And we thought that we can easily make the conclusion that from where this pathogen came and its genetic identity. But practically, it was not so easy, because when we got the data, we checked the NCBI gene bank, we found only one isolate of BR32 genome sequence are available. And this is why we changed our strategy. What we did, we made a call to the global scientific community. Scientists issue rallying cry for wheat blast research, and we released all of our data through Open Wheat Blast website as it was released from the machine. We thought that many scientists globally who are working on wheat blast, they have the sequence data and they download and compare and analyze the data. And finally, here are the 12 samples, the original data we shared. And it was supported by the Nature. They also supported our wheat blast, promoted, and science also promoted our wheat blast so that global scientific community can be engaged. And fortunate very rapidly. Within a couple of days, many scientists from UK, Switzerland, France, Germany, South America, they download our data. And finally, we combine all the data and we found that Bangladeshi wheat blast originated from the South American wheat blast because they are very close phylogenomically. And here you can see within 6 weeks of time, we shared our data through bioarchive, that is a preprint. Here you can see people participated, and here is our newspaper, and more than 50 media globally shared this story. And within 6 months of time, we published this paper in a flagship biology journal, BMC Biology, and it is the highest cited paper in the history of wheat blast. That is, emergence of wheat blast in Bangladesh was caused by a South American lineage of Magnaporthe oryzae. Unfortunately, wheat blast came possibly through a grain import from the Brazil, and they found the heaven in Bangladesh. And here you can see 31 researchers from 4 continents they joined. Just 1 year later, as we got the report from India, Hindustan Times, leading newspaper, that India also, West Bengal, 2 districts affected by the wheat blast. And India declared wheat holiday, no cultivation of wheat for 3 years in West Bengal. They took the nice decision. Unfortunately, just 2 years later, Zambia, an African country, they also reported the wheat blast, and we collected the samples from Zambian, isolate and Sophien, and we international community. We analyzed and we found that Zambian one was also originated from the South America. There are lots of study to understand the vulnerable area of wheat blast. A large area of global wheat producing country, the environment is very favorable to wheat blast. In case of wheat blast, the biggest problem is there were no precise and convenient diagnostic method for wheat blast. This is why it is #1 export-import crop. So there is high chance to move other countries also. Another problem is resistance sources are very limited. Classical breeding is very time-consuming. Even genetic engineering is very complicated in case of hexaploid wheat. Fungicide are ineffective and hazardous to the environment. Especially wheat blast is head disease. In the vegetative growth, you cannot see the major symptoms. So when head neck is affected, then you have nothing to do. So to tackle this terrible disease, we decided to utilize the CRISPR-Cas technology, the smartest technology discovered by two lady, Professor Emmanuel Charpentier and Jennifer Doudna. They got the Nobel Prize in 2020, should not be 2022. And by this technology, people started to use in many aspects of genome editing of many crops, and it has already been established by many cases to pinpoint and precisely detect the disease. And we also decided to first develop a uh, precise and point of care diagnostic method before having such a smart diagnostic method. I collaborated with Professor Guliang Wan and Chinese Academy of Science and USDA scientists. First, we discovered two genome-specific primer in the genome of Magnaporthe or oryzae triticum, that is wheat blast fungus. And these based on this, nucleic acid fragment, we designed the primer, and first we developed the PCR technique. But PCR technique you cannot deploy in the field, or even not good for the quarantine offices. Then we upgraded the method into loop-mediated isothermal amplification. No need PCR, but you need read at temperature, and it is also smart method and easier. But we thought to make more convenient. Then we utilized Cas12 enzyme, and because we targeted the nucleic acid fragment to primer and Cas12 enzyme we utilized, and this PCRD strip very smart and very rapidly you can determine. Here you can see in case of wheat blast, you can find the single band, and in case of negative 2 band or uncertain or invalid like that. So this strip we can use to detect wheat blast 30 minutes, and it is the smart nucleic acid based technique. And we already made a contract with an industry to utilize this technique and making it cheaper and user friendly. And to improve this method, we also collaborated Magnaporthe because he developed bioscan for detection of virus COVID-19. And this is more smarter than the our one. Then we collaborated with him and upgraded this, CRISPR lateral flow assay. And that method can be not only be utilized for Magnaporthe oryzae, just changing the guide RNA, you can detect any fungus. We recently published a paper in Plant Biotechnology Journal thanks to Magnaporthe. So here is the main flow chart of how we can do the smart detection of wheat blast utilizing the CRISPR-based lateral flow immunoassay. And by changing the guide RNA, it can be robust technique for detection of any nucleic acid fragment, even in the DNA, GMO, or even in the cell. or even genotyping or marker-based breeding method, and obviously virus, bacteria, and fungal diseases. And after development of that technique, our major target was how can we develop a durable blast-resistant wheat variety. We thought that CRISPR technology could be utilized. Sophien and Nick visited Bangladesh, and we targeted to edit the S genes, that is disease-susceptible genes in the wheat genome. Actually, wheat genome have been sequenced and annotated in 2018, and it is a hexaploid, very complex, and most of the genetic engineer keep it aside. But we decided to utilize this technique because Sophien is champion in utilizing the CRISPR-Cas technique in plant. Jennifer and Emmanuel, when they discovered in 2012, Sophien utilized it in 2013. So we decided to edit some of the rice orthologous gene in wheat susceptible gene for the blast, because in some cases, some of the experiments in other diseases, people found that editing of the S gene can, make the plant immune. And to prove the concept, we first used the barley, and it worked very well. And lots of, guide RNA have been designed, and finally 10 S are edited, and this CRISPR construct available in the Ed gene, and we found the lots of 7,000 lines, and we screened them, but we found moderately blast resistant, because when we check the sequence, our method not so precisely edited the S gene. In some cases, a big segment was edited. It's very difficult, and we have to optimize the method. That was one story. Another story is that there are two bona fide genes discovered by Professor Yukio Tosha group, RMG8 and RMG-GR119. They are resistant against the wheat blast. And other one, 2NS translocation, have been deployed in wheat breeding in South America. But these two new genes have never been used. In Bangladesh, 2NS translocation is deployed. We intend to deploy these two genes progress into our 2NS translocation and other good varieties. That would be fantastic. And thanks to Yukio Tosha, because he offered us the genotypes, and we integrated, and we found, now F3 and F4 population, backcross population, several lines we found very promising candidate. We had another approach to utilize the probiotic bacteria, endophytes, from the seed of wheat for biocontrol of wheat blast. We found a good number of bacilli, a combination of them, consortium of them can, at field level, can control the wheat blast, and possibly through different chemical producing by the bacilli and enzymes, antimicrobial, as well as induce the systemic resistance. And we identified some of the pure compounds also. And not only biocontrol, but we also try to utilize the nanotechnology, nanopesticide. Professor Yusuke Yamauchi is one of the famous nano architect, and we designed a nanocatalyst, titanium dioxide, which is, in the daylight, in presence of oxygen and moisture, it can produce the reactive oxygen species at certain concentration. If we use in the field, then it can kill the fungus, but not the plant. Plant has thick cell wall, and it is fantastic. And in the field level, we found very nice. We also collaborating with mutation breeding, International Atomic Energy Agency funding us, and also Institute of Crop Science of Chinese Academy of Science, and we found many mutant lines generated by gamma radiation, ion beam, X-ray. They are promising candidate for blast resistance, and we are going to do genotyping. Here is the recent blog post in the Medium by Sophien Kamoun, October 7, 2022. He made a question: could a new threat to cereal production be emerging in Europe? Because they found in Germany from foxtail, millet, and crabgrass, the Magnaporthe oryzae, which can even in the laboratory condition infect the wheat. And he made a warning so that there is whether there is any chance of threatening wheat production in the Europe. I am very fan of the open science as well as open data sharing. Based on my experiences, it has lots of advantages. In case of our wheat blast work, we had no funding, but as all the scientists willingly joined, then we solved it very nicely. Here is our experiences, how South and West scientists work together and finally utilize the open science to rapidly tackle the serial killer on the run, that is wheat blast. And in Bangladesh, pinpoint of the origin of that disease helped us to make an international workshop and invite all the South American scientists and make the policy decision, and that helped us to tackle future epidemics of wheat blast. So in case of open science and open data sharing, there are advantages and some challenges also. So how we overcome, you can find our thoughts in the PLOS biology paper. So I am at the end of my talk. What should be the take-home message for all the audience? The oryzae wheat killer fungus Magnaporthe oryzae does not sleep, and it is on the run because it is already in the 3 continents. And we urge global scientific community to join our fight against the wheat blast fungus. We must find a solution, solid solution, before it becomes catastrophic in Asia, because China and India, Pakistan, they are the largest wheat producing country. China #1, India I mentioned second in the world. And if wheat blast is there, then it would be big problem for the world for food security. Open data sharing and open science are fantastic approaches for rapidly tackling any plant health emergencies. Not only plant health emergencies, any emergencies related to the mankind, we can work together. And I must acknowledge the collaborators, especially the Cartoon and Reem, Kaixia, who are leading the wheat blast initiative pathogen control. They offered me opportunity to talk here, and lots of outstanding globally leading scholars who have been supporting us for wheat blast research. I am very much thankful. In my team, and now around 15 people, postdoc, PhD, masters, they are working. That is, wheat blast not only infected Bangladesh, my whole team dedicated to fight against the wheat blast, and we are receiving funding from many donors, and we are highly thankful to all the donors. And my students and team members, they are very dedicated. Thank you so much from my team, and we welcome international collaboration and support to win the fight against a trivial fungus, Magnaporthe oryzae critica. This is all about my presentation. I shall be very happy if you ask question or make any comment.
Thank you very much. That's fantastic. Very nice talk. Thank you. I see Karchan has his hand up. Go ahead.
Hello, Tofazzal. Hello, Reem. Great talk. I really love your talk, and thank you very much for providing such a nice talk for us. Kachin here from Curtin University. I think first of all, I would like to say that the little COVID-like test that you've got going, that's pretty neat, and I guess some of us would like to sort of move that direction one day. But I think in terms of the aspect that you mentioned, that a wheat blast itself, that fungicide control against wheat blast itself is quite inefficient. Would you be able to comment a little bit more about that? Why is it inefficient, and has different formulation been tried? Why hasn't really been hasn't fungicide working out for the wheat blast community?
Thank you so much. It is a very nice question. Actually, in case of fungicide, first we used fungicide to treat the seeds. We thought that it is seed-borne, so a seed should be clean from the fungus. But next year, seed treated with fungicide and seed without treated fungicide gave the same disease, because in case of wheat blast, it is not yet been precisely understood the life cycle from where the conidia comes. We checked the first rain. We found the DNA of the blast fungus. So it is one problem. Second problem is when farmer use the see the blast symptom in the head. It is particularly head disease. When whitehead symptom as farmers see, then if you apply the fungicide, you can kill the fungus, but cannot save the crop, because it infects the neck of the head and block the vascular system. So already blocked water supply as well as nutrient supply for the growing grain is problematic. But if grain is filled in at that time, if infect, then damage is very low. But if just at the beginning of flowering, mostly it attacks at that stage or milking stage of the grain filling, then it is not so effective. But fungicide in the laboratory, you can control it. And as symptoms are not visible in the vegetative stage before flowering, on the leaves, it is a very big problem whether your field has any fungus or not. This is why we developed this smart point of care detection method, so that earlier in the field, if you find the fungus is there, then people can use the fungicide. There are some fungicides.
Okay, thank you.
Very interesting.
Thank
Thank
you.
you. That's very good. Any other question? I actually have one. This is very interesting topic for me. I find it quite fascinating how the disease is spread by, like, basically a seed, right? Came through South America to Bangladesh and can devastated almost threaten whole India and Bangladesh. That's kind of scary. I was wondering what your take about it, that now in Germany they are trying to detect it, in Europe, etc. Those places where are known to be more of climate to the cold, we almost like in Canada, we feel like in 2016, I remember talking to my colleagues, and the often reply I would get, oh, don't worry, this is just only in warm countries. But pathogens do adapt actually to climate and stuff. So what do you think? What's your take on that? And the other thing, any measurement to, like, in terms of quarantine, like did the Wheatoplast change way of how you guys import seed or anything about regulation wise? Would you kind of clue us to that?
Excellent question. Yes. In Bangladesh, we cultivate wheat in winter, and winter is dry season. And in 2016, 8 districts experienced unexpected rainfall in February and heavy shower. And that 8 districts devastated. And we studied thoroughly the climatic requirement, that is environmental requirements of the wheat blast fungus. South American experiences and our experiences matched. It needs 18 to 13 degrees Celsius and high humidity. High humidity may be through dew falls or rainfall. And these are must, because leaf must be wet for 30 hours. Otherwise, no epidemic would be happen. You can see minor infection. As climate is changing, especially meteorological parameters, seasonal meteorological are not stable. That tradition is breaking. And wheat blast in Bangladesh devastated the wheat within a week. If the favorable environment is available within 3 or 4 days, it can proliferate exponentially and devastate the wheat. In case of Europe, actually the weather is not so favorable for the wheat blast. But the pathogen, the Magnaporthe oryzae, it is a very complicated, pathogen. It has transposon elements and can mutate very fast. In South America, 200 races are there. So it is difficult to predict whether wheat blast would outbreak would be in Europe or similar temperate region or not. But if a host germ happens from the grass to the wheat, those are adapted over there. And if host germ happen, then it may be a problem. So at this moment, it is hard to predict, but we should be cautious enough. As Sophien mentioned, in the laboratory condition, he found, millet as other grass, Magnaporthe oryzae can equally infect the wheat. But it does not mean that in the practical field it would be similarly happen.
Yeah. Yeah, that's very good. Well, I see a hand up for Marcel from, I think, from South America. Yeah?
Yeah. Can you hear me?
Yes, we hear you.
I don't know. I don't know if you are seeing me. But my intention is that you see me. My English is not so good, and I would like to say that it's a pleasure to talk with you. And here in Brazil is where this disease was born. Let's say this way. And I missed some part of Dr. Islam presentation. Sorry if my question is not connected, but I would like to know something about the experience in Bangladesh in relation this sequence 2NS in relation to the resistance as source of resistance to wheat blast. Here in Brazil, let's say that we are living a situation that we have some success with this strategy. I would like to hear something about this, your experience there in Bangladesh in relation to this sequence. Thank you.
Thank you.
Excellent, Marcel. A nice question. Actually, Bangladesh was fortunate when Bangladesh experienced wheat blast. At that time, a wheat variety was enriched with zinc in the grain, a high zinc content in grain, ready to release. But when wheat blast origin was determined, that we decided to send the variety to Bolivia to test whether it is blast resistant or not. And it has been found that it contains 2NS translocation, a chromosomal segment, as Michel mentioned, which is the blast resistant. And we deployed in our field, and we found that, yes, even in the very good environmental condition, it can protect the plant about 70 to 75% or like that, but not 100%, resistant. This is why we are trying to introgress RMG8 and RMG-GR119 in that background, because in Bolivia, 2NS translocation was override by B71 strain. So there is a little bit risk, but still 2NS translocation in our farmers field showing very good resistant against the wheat blast. Thank you so much. Very nice question.
Okay.
Thank you. Thank you. That's good. I see Chloe Rodisha.
Okay.
Yeah.
Hi. Good evening to you, Dr. Islam. Thank you for the presentation. So my question is, so it was very good that you already have the product of like the kit, like the COVID kit, right? So and then you did mention that you already contract out with companies. So what is the status now of that plan? Because it is very good. It would be on-field system. then I do believe that there will be a recording system so that we can pinpoint out in the world also who are this. And since it would be farmers, how much do they have to pay, or is it a government program in a global way? Because at the end, that's the product that we want to. And at the same time, when we will have this wheat resistant ones, we will see the end product of us fighting against this problem. Thank you.
Yes, excellent question. Yes, our CRISPR-based lateral flow immunoassay, we are also very serious to deploy it in the real field, quarantine offices as well as in other countries in the farmers field, and this is why we made an investment in
Uh-oh.
giving us the faster response. So our collaborator in China, they communicated with some company and they asked us to make huge copies, but you have to pay per a piece of diagnostic kit $5.5, and it is also expensive. This is why Food and Agriculture Organization, they communicated with us to deploy it in the farmers field, and one project is on process. Another good news is that Bill and Melinda Gates Foundation, who is funding the Global Rush Project, Cornell University and CIMIT, they are involved there. And they included us in that project for, blast. Sufyan and we have two sub-projects, and that fund has recently been approved. So we hope we can utilize those funds to deploy this technology in the real field so that we can stop the further spread of wheat blast as well as monitoring and surveillance of this pathogen in the field. Thank you so much. Very nice question.
Thank you, Rodisha. That's really good, the question. Okay, then it's just, I see, let me see if I see, just a second. I'm gonna see where the people are. I don't see any other hand. I just one comment following up on the quarantine question. I was hoping to get an answer to that. Like anything it change in terms of measure to how import seed or anything like that? Did the blast lead to any changes regulation wise?
Now, this is interesting question. Actually, Bangladesh didn't import wheat seed, actually grain, food grain. But in case of wheat and rice, there is a difference.
Okay.
Even if you bring food grain, and if that grain touch the soil, then it gives the plant. In quarantine offices, as our method is practically validated in the field, it can be deployed. That would be fantastic to check when a grain import is moving, whether there is blast or not. In case of Zambia and Bangladesh, in both cases at that time, there are no smart, technique for detection of wheat blast. That was the reason. But now we have the technology. Important is how can we deploy the technology in the field level.
That's very interesting. Well, that's really treat for today kind of
And
talk.
another point is that another point, I'm sorry to interfere. Another point is that wheat blast fungus, it is a biosafety level, 4, I think, because it is the deadliest pathogen. So in case of wheat export import, the regulator should think about confirmation of whether the grains are or seeds are free from wheat blast through a smart method, I think. Thank you.
Thank you very, very much. Thank you. Well, if we have no other thing, I guess I'm gonna stop the recording, so it will hopefully we'll share it on the website. And